Hi, welcome to Shruti Tutorial YouTube channel. Now we will learn Southern blotting experiment. Aim: to detect the presence of a specific DNA sequence within a complex mixture of DNA using a labeled probe. Materials required: DNA sample, for example, genomic DNA, restriction enzymes for DNA digestion, agarose gel and jelly electrophoresis apparatus, buffer solution for electrophoresis, nylon or nitrocellulose membrane, hybridization buffer, labeled DNA, RNA probe radioactive, fluorescent or enzyme-linked, autoradiography film or other detection equipment, for example, fluorescence reader, vacuum transfer or capillary transfer setup for DNA transfer to membrane, heat source for DNA denaturation, for example, heating block, UV light source optional for visualization of DNA bands in the gel, plastic bags or hybridization chamber for hybridization, washing buffer to remove unbound probes. Steps: preparation and digestion of DNA. Extract DNA from the sample and digest it using specific restriction enzymes to produce fragments of varying lengths. Jelly electrophoresis: separate the digested DNA fragments by size using agarose jelly electrophoresis in an electric field. Small fragments move faster and farther through the gel matrix compared to larger ones. Denaturation of DNA: soak the gel in a denaturation solution, usually alkaline, to convert double-stranded DNA into single strands, allowing for efficient probe binding. Transfer blotting of DNA: transfer the single-stranded DNA fragments from the gel onto a nylon or nitrocellulose membrane. This can be done by capillary action using absorbent paper or electrophoretic transfer. The DNA binds to the membrane, replicating its position in the gel. Hybridization with labeled probe: prepare a probe complementary to the DNA sequence of interest and label it for detection, for example, radioactive isotopes, fluorescent dye, enzyme. Incubate the membrane with the labeled probe in a hybridization buffer at an appropriate temperature, allowing the probe to bind to its target DNA sequence on the membrane. Washing the membrane: wash the membrane to remove excess and bound probes, ensuring only specifically bound probes remain. Detection: detect the bound probe using an appropriate method based on the label, for example, autoradiography for radioactive probes, fluorescence detection, chemiluminescence for enzyme-linked probes. Analyze the resulting bands, which indicate the presence and size of the DNA fragments complementary to the probe. Significance: gene mapping and identification. Southern blotting allows for the identification and mapping of specific genes within a DNA sample. Diagnosis of genetic disorders, used for detecting mutations, deletions, or rearrangements in genes linked to specific diseases. Forensic science plays a crucial role in DNA fingerprinting for criminal investigations and paternity testing. Molecular cloning verification confirms the presence and correct insertion of DNA fragments in vectors,